Hello and welcome to today's video on know more about malaria. In today's video tutorial, we will be covering some of the important questions related to malaria, which are not usually covered under syllabus. The questions like why malaria is mostly found in Africa, why cell heterozygosity, that is sickle cell heterozygosity, affects mostly African people despite its ubiquitous nature. We will be knowing how hemoglobin C protects against severe malaria, why fetal hemoglobin provides resistance against malaria, how alpha thalassemia protects against severe malaria, why glucose 6 phosphate dehydrogenase deficiency is found mostly in males of malarial endemic areas, or why and how Duffy antigen provides protection against Plasmodium vivax, or which glycophorin proteins provide resistance to the invasion by Plasmodium falciparum. In this video, we will be trying to cover in detail the answers to these very questions. So first of all, let's try to know why most of the malaria cases occur in Africa and why the death rate is high there. Malaria, as we all know, it's a disease caused by a causative agent, that is Plasmodium, and female Anopheles mosquito acts as carrier or transmitter of this very causative agent. The disease can be life-threatening. An estimate based at global level that this disease killed roughly 4 uh, lakh 9,000 people in a single year of 2019 alone. Globally, an estimated 3.4 billion people in 92 countries are considered to be at the risk of being infected with malaria and developing symptoms. It is quite surprising in this case that most cases occur in African continent, in sub-Saharan region particularly. It is because a particular type of mosquito, that is the Anopheles gambiae complex, that is responsible for high effective rates of transmission resides there. The local weather conditions there allows for the transmission to occur round the year. Plasmodium falciparum that causes the dangerous malaria. This species is endemic to this African region. Limited resources and socio-economic instability, that is poor conditions of the countries in sub-Saharan region, and it also makes the malaria control difficult there. That's why malaria causes most of the deaths in African region, and it is mostly found there. Now let's try to know why sickle cell heterozygosity affects mostly African people despite its ubiquitous nature. Sickle cell heterozygosity. It is a hereditary disease. It is characterized by an abnormal structure of the hemoglobin molecule. In the hemoglobin molecule, where glutamic acid on position 6 on the beta chain in hemoglobin is replaced by valine. This minor substitution has profound physiological implications. Under low oxygen conditions, stacking of these sickle hemoglobin molecules occur and distortion of the RBC membranes results forming a sickle shaped cell. This is called sickle cell heterozygosity. It is a recessive autosomal disease. That is, this disease show recessive autosomal transmission. When the sickle cell condition is found in homozygous recessive state, it leads to the death of that very individual. However, the heterozygous condition in which one gene is normal and another gene is showing this sickle cell trait, this is believed to confer a degree of resistance to malaria. That is, in simple terms, we can say sickle cell heterozygosity provides a degree of resistance to the malaria. In other words, we can say that sickle cell heterozygosity gives an advantage in malaria endemic areas. The possible reasons for this may be: one, cellular adaptations. In cellular adaptations, we see that the RBC of a sickle cell carrier individual, when somehow it is affected by the plasmodium, it shows less adherence to the vascular endothelium. Also, the RBC, this RBC with sickle cell heterozygous individual, it shows a weak expression of Plasmodium falciparum erythrocyte membrane protein on its surface. For a Plasmodium falciparum to infect an RBC, there is the requirement of expression of a protein on the surface of RBC. And when the RBC shows less expression of that very protein, so naturally this Plasmodium falciparum will not affect or infect that very RBC, which shows a less expression of that very protein that is required for this Plasmodium falciparum to attach with or to infect that very RBC. Another possible reason may be biochemical adaptations of RBCs. Uh, During sickling, there is potassium loss, and the osmotic loss of water results from the loss in potassium. It results in the increase in concentration of hemoglobin, sickled hemoglobin HbS in the cell, as well as its polymerization. That is, loss of potassium from the cell increases the loss of water from the cell, and the loss of water from the cell, it enhances the polymerization of sickled hemoglobin inside the cell. And these two phenomena, that is the loss of water and sickling, uh, they are the main determining factors that inhibit the growth of a parasite inside the cell. In addition, on infection to RBC, Plasmodium targets hemoglobin and destroys it. This reduces oxygenation, and this reduced oxygenation leads to the deforming of the cell due to the polymerization of HbS as already discussed. These deformed RBCs are repeatedly cleared from the body. So the parasite clearance occurs along with clearance of deformed RBCs. Since the non-parasitized RBCs are dominant, the individual's chances of survival increases because the malarial parasites are regularly eliminated along with these deformed RBCs. And the point should be noted that these deformed RBCs are normally not in a deformed state. Rather, the targeting of the hemoglobin by this parasite results in the stacking of these HbS due to the loss of water and potassium, and this results in the deforming of the RBCs. And as the malarial parasite has already entered this RBC that is deformed, and the clearance of the deformed RBC, it results in the clearance of the parasite as well. Why fetal hemoglobin provides resistance against malaria? It is the question. The adult hemoglobin structure is alpha 2 beta 2 chains, but the fetal hemoglobin structure is alpha 2 gamma 2 polypeptide chains. That is, the fetal hemoglobin contains a gamma polypeptide chain in place of beta chain of hemoglobin A. Hemoglobin F contains 2,3-diphosphoglycerate. That is, hemoglobin F binds 2,3-bisphosphoglycerate, and which has a low affinity for oxygen. Uh, this low affinity for oxygen inside the fetal RBC makes unfavorable conditions for the parasite Plasmodium to grow there. Now let's know how alpha thalassemia protects against severe malaria. As we already know, hemoglobin is a tetramer made up of alpha 2 and beta 2 chains. When defects in alpha globin chain genes results in decreased synthesis of alpha globin chains. That's called alpha thalassemia. And when the defects in the beta globin genes results in the decreased synthesis of beta globin chains, it is called beta thalassemia. Malarial protection because of the alpha or beta thalassemia has been a very controversial topic, because there are number of reasons given in this respect, that how this alpha thalassemia or beta thalassemia protects against severe malaria. In thalassemia, it is generally believed that oxidative stress plays a protective role, because less alpha or beta globin chains leads to the less oxygen binding, and this creates oxidative stress inside the RBC. This oxidative stress inhibits the growth of RBC inside the the growth of parasite inside the RBC. This might be one of the reasons. Some studies have also indicated that Plasmodium infected alpha thalassemia erythrocytes. That is, the erythrocytes are alpha thalassemia. Alpha globin chains are secreted less in these RBCs. They bind high levels of antibody sera from malaria endemic areas. That's what alpha thalassemia erythrocytes have shown high levels of binding of antibodies from the malaria endemic areas. As we know, antibody binding leads to the immune response at high levels through opsonization, through complement mediated lysis or antibody dependent cytotoxicity, etc. We can say that this alpha thalassemia erythrocytes, this enhanced immune response in these can be one of the reasons for the protection against severe malaria. Now it will be quite interesting for us to know why glucose 6 phosphate dehydrogenase deficiency, that is G6PD, is found mostly in the males of malaria endemic areas. As we know, G6PD, that is glucose 6 phosphate dehydrogenase, it plays an important role in the reduction of oxidants, and which are indispensable in cellular metabolism and in RBC survival. That is reduced oxidants, that is the reduction of oxidants. It is needed for the cellular metabolism and RBC survival. When there is the case of glucose 6 phosphate dehydrogenase deficiency, the oxidants will be rich there in RBCs, and oxidants richness, it provides less favorable environment for the development of the parasite. That is G6PD deficiency in a patient, it leads to an advantage against the malaria. Since this G6PD gene is found on the X chromosome, and in the males there is single X chromosome, this G6PD gene or this G6PD deficiency provides an advantage in malaria endemic areas, so it gets selected during evolution. So we can say that this G6PD deficiency is found mostly in the males of malaria endemic areas, that is particularly Africa. Now let's know why Duffy antigen provides protection against Plasmodium vivax. The Duffy antigens are actually chemokine receptors. Chemokine receptors. These are antigens expressed on the surface of RBCs or other cells of the body. The Duffy antigens are uniquely recognized by Plasmodium vivax for its infection to RBCs. That is, Plasmodium vivax is unique in human malaria infection in that its invasion on the erythrocyte depends exclusively on the presence of Duffy antigen on the cell surface. That is, if Duffy antigen is present, the Plasmodium vivax will be able to infect that very RBC. It is possible that some genotypes may show no expression of the Duffy antigens, and this genotype where no Duffy antigen expression is present confers complete resistance to Plasmodium vivax infection. This genotype is very rare in European, Asian, and American individuals, but it is found to be very frequent in populations of indigenous West and Central Africa. It is thought to be due to the very high exposure of Plasmodia, very high exposure to Plasmodium vivax in Africa over the past few years. It's also quite interesting to know that blood group O provides some protection against severe falciparum malaria. This protective effect of blood group O and susceptibility of blood group B to severe disease. It is quite interesting, but the studies are still on, and it is to be known fully how blood group O provides protection, whereas the blood group B is quite susceptible for the severe disease of malaria, particularly caused by the Plasmodium falciparum. At last, let's know which glycophorin proteins provide resistance to the invasion by Plasmodium falciparum. The glycophorin A, the glycophorin B, and the glycophorin C. The erythrocytes which are deficient in glycophorin A, glycophorin B, and glycophorin C, they are resistant to the invasion by Paris Plasmodium falciparum. That is, the erythrocytes where the expression of glycophorin A, glycophorin B, and glycophorin C is nil. They are resistant to the invasion by Plasmodium falciparum. These were some of the quite interesting protective mechanisms that are getting evolutionary advantage, particularly in malaria endemic areas, and somehow it is leading to the change in the genetic makeup of the individual. That is, the evolution of human genes is also happening because of this malaria, particularly in malaria endemic areas. Hope you enjoyed the video, and hope you learned quite interesting and many new things about malaria. Thanks.
P Thank you. Jazakallah.